Good morning, everybody. We have a very intense day today. Everything is happening here. All the events are happening here, beside, of course, the evening departure to an interesting restaurant with music. We'll start with Aya Ben Yakov, who did her first degree at the Hebrew University in computer science and cognition. Then Aya moved to the Weizmann Institute to do her PhD with Yadin Dudai. Aya is a cognitive neuroscientist, and she went to Cambridge for her postdoc, and then we were lucky to get you back. So it's your stage, Aya.
Thank you, Dan. It's very nice to be here. Thank you very much. Quite excited. And I'm going to share our work on memory. And to start, I'd like to ask you... Oh, maybe I'm not holding it close enough. Okay. So I'd like to ask you all to think back to your morning, let's say maybe to breakfast. And when you think back to breakfast, maybe you remember yourself kind of looking at the breakfast buffet, trying to decide what you're going to get. These are called episodic memories. These are the memories that Chaim talked about yesterday. They're memories for our own experiences. Memories where you can remember yourself as part of the event. It's as opposed to knowledge, which is called semantic memory. And these are the kinds of memories that we're interested in understanding. We want to understand how they're formed and what they include. It's something you would have thought, okay, we already know what these memories are. They're so interesting, but we still don't really know what they are. And of course, they're so important scientifically, culturally. We can really think of memory as the architecture of our identity. If you think back to Chaim's talk, when he wants to create a biologically inspired AI agent, the first thing that agent needs to have is episodic memory. That's such a big part of our identity. And I like showing part of a movie from Inside Out, which, have you seen this movie? Some yes, some haven't. So this is part of the movie that talks a lot about episodic memories.
I don't want to get too technical, but these are called core memories. Each one came from a super important time in Riley's life, like when she first scored a goal. That was so amazing. And each core memory powers a different aspect of Riley's personality, like Hockey Island. The point is, the islands of personality are what make Riley, Riley.
So these are really kind of what make us who we are, in a sense, these memories. But how can we study this memory formation? When we think about it, if we want to study these memories, we can't study them in animals in the same way. We can study things that are like these memories, but it's a bit like the philosophical zombies that Leon was talking about. We can study how animals behave, how they respond, but we can't ask them what they experience, what they remember. So how can we start studying this in humans? For a very long time, the main approach people had to studying cognition in humans was to see what happened when things went wrong. Let's say if someone lost his speech, the scientists could then check which brain region was damaged and say, okay, that's the speech region. But there was a problem because no region for a very long time seemed to be related to memory, and this amusing quote demonstrates it. I sometimes feel in reviewing the evidence on the localization of the memory trace that the necessary conclusion is that learning is just not possible. Right? This is kind of a scientific approach. We can't find it, so it's not real. Nevertheless, in spite of such evidence against it, learning sometimes does occur. And then this all changed when there was an unfortunate case of someone called Henry Molaison. He was known as HM. He was operated on for epilepsy. And from that moment on, he couldn't form any new memories. So every morning, these doctors he would meet over and over again for years, every morning. And I apologize, this may have happened to me with some of you yesterday. He'd say, oh, hello, nice to meet you. My name is Henry. Walk out the door once again. And there's a nice movie, Memento, which gives you kind of an inside look into what this experience is. And when they looked at the brain region that was damaged, this led people to understand that a region deep inside the brain called the hippocampus was the one responsible for memory. Without our hippocampus, that Chaim also mentioned yesterday, we can't form these new episodic memories. And it's called hippocampus because it seemed to resemble a seahorse. And that's why this is its name. Okay, great. So now we know the hippocampus is responsible for memory. But we want more than that, right? We want to understand how these memories are formed. Ideally, we want to look inside the human brain and see what's happening when these memories are formed. And luckily for us, came the invention of MRI. MRI machines use an extremely strong magnet to enable us to image the brain, other regions of the body as well, but we can get highly detailed regions of the brain. And then an additional development, functional MRI, enables us to take every point in the brain and look at the brain activity in that point in the brain over time with amazing precision. It's not direct brain activity, it's this very innovative approach of looking at blood flow, because when a region is active, you send more blood there. So it's not a direct measure, but we can actually look at brain activity in healthy humans as they're performing tasks. And then we can look and see these maps and look all over the brain and see which regions are involved in something that interests us, let's say memory. And we can get these kind of plots that show us if something is happening here at the vertical black line, what the response looks like in that area that interests us. It can be stronger or weaker. And one of the first things researchers did was try and look at how we form memories. This was one of the kind of quests to understand. But how can we do that? So there's something called a subsequent memory paradigm, where we look at brain activity that predicts what's going to happen later. What they did was they put people inside this MRI scanner and they showed them words, adjectives like dirty or happy. And before each word, they had this instruction. Either imagine a place that word describes or read it backwards. Okay? And then they test memory for these words. So now the idea is, if we see that there are areas in our brain that are more active when you're looking at something that you're going to remember, so I can look at your brain activity and tell you what you're going to remember and what you're going to forget, that means we're seeing the formation of these memories. And so they looked at this. Here, these bars show you the overall activity in the hippocampus. They looked at those words that were later recognized as being seen or later forgotten, and they see nothing in the hippocampus, which is strange. The hippocampus is involved in memory. No difference, yes. No difference between a word that's remembered or forgotten. But when they then test here whether for each word it was under the instruction to imagine a place or read it, now they suddenly see a big difference between this red bar, those words where they remembered what they were doing, and those words that they didn't. And this gives us another important clue that the hippocampus binds together memories. It's not just a memory for a single word. It takes together different elements of memory, and it's the one that's responsible for putting them together. And this will be important as we go along. These were images in this case, but if they hear the words, it's the same. Or if they see pictures, it's the same. But note that in all these examples, our real memories are kind of different, right? If you think back to your memory of the breakfast, that was a bit different from these. Oops. And to demonstrate, I'd like to share one of my own memories. This is a memory of my daughter, Yara. Okay. So this is some occurrence sometime, someday.
Superman.
So So for some reason, she thinks Batman is Superman and the Superman roars. But in any case, this is a nice memory that I have. And it's very, very different from remembering the word happy. And this is what we want to understand. How do we form memories for these kinds of experiences? And Daniel Kahneman makes a very important distinction between our experiencing self and our remembering self. We tend to think that we go through life, we experience things, and then we remember them, right? It seems trivial. But what we experience, the self that is experiencing things, is very different from the one that's remembering. What do I mean? The experience itself is a flow of moments, right? We see frame after frame after frame. That's our experience in life. It's like this continuous movie that you just get single snapshots one after the other. But if this is like a movie, our remembering self, the way we remember these experiences, this is like a trailer of that movie. It contains the story. It's not single moments, but the story of what happened. So I remember the sequence. She wanted to jump. She was afraid. She walked down. Then the Superman and the roar. And these trailers, this is what we want to understand. How do we create these stories that form our lives? So how do we go about doing this? So what we did was to use something similar to the experiment I described. We put people in a scanner and we showed them clips instead of showing them words. So these clips that evolve over time, they're a bit more like our real experiences. The original clips were in Hebrew, but I'll give you an example of what this looked like with clips in English.
We've definitely lost the trail. I think we veered off around that last fork by the ridge. If we backtrack now, we'll be walking.
Oh, yeah, there are some issues between.
I think there was some mix-up, but I ordered a sandwich and orange juice.
Okay, so these were the types of clips that we showed. And then we want to test memory. We asked what happened in the scene, and now we can look at the brain activity to scenes that were remembered and scenes that were forgotten. But then, as I was preparing this, I felt that I really needed, you notice there was this gray screen in the middle between scenes. So that is there for technical reasons in fMRI. But I felt I needed that. I couldn't see 160 clips without pausing between them and manage to remember anything. I needed those breaks. So I thought, okay, maybe if I need the breaks, my brain does too. So we also looked at what happens at the end of those clips. And this was very serendipitous, as you'll see in a moment. So when we look at the activity during the clip, and we compare those clips that you will later remember and later forget, we see a lot of different regions that are similar to what people see. But what was really interesting was what happened when we looked at that activity at the end of the clip. We saw very few regions, and I'm going to focus on this now. So these are the regions. One of them is called the striatum, which is a region usually involved in habits. One is the cerebellum. And to our utmost surprise, we found the hippocampus. So we found it waits. If we look at the activity here, this is the activity in the hippocampus. I'll cover the duration of the clip. This looks just like these responses I showed you. This looks exactly like a response to the end of the clip. That's stronger if you're going to remember. So if I look into your brains, at the hippocampus, at the region involved in memory, and I look during the clip, I don't know if you're going to remember it or not. But if we wait until the end, and I look at your brain activity, I can tell you if you're going to remember that clip. And if I show you a longer clip, the response is going to be later in time. We showed that. We use clips of different lengths. So here I'm focusing on the hippocampus, but we're also now trying to understand what's going on in the striatum, some very interesting things there, so stay tuned. Maybe in the next brain circle, we'll talk about that. But what's an end? If we look at these clips, we have a clip and then a fixation, this plus in the middle, where we tell people to look at that. is the hippocampus responding really to the end of the clip, or does it like it when you have pauses, right? When you just stop everything and you rest. So here we added 2 clips, one after the other, and we want to see what happens. We know what's going to happen when we have a single clip followed by a break. We already saw that. But for 2 clips, we have 2 options. One is that the hippocampus likes fixations. It waits until we don't have any information coming in. And the second is that it likes things to be wrapped up at the end of something. So if it likes fixations, it's going to wait until both clips are over and then respond. And if it likes ends of clips, it's going to respond at the end of each of the clips. And we're expecting a response like this 2-peak response. And this is exactly what we get. So we find that the hippocampus really responds at what it considers the end of something. Okay, so now we're on a quest to understand what that end is. Yeah. Alzheimer is a much more complex memory phenomenon. The hippocampus is involved in Alzheimer. It's certainly a region very much affected, which is why our memories are so affected. But the pattern is a bit different. This is more, you see this more involved when people have the problem to form new memories. But I will mention how this can be related to disorders at the end as well. And you also want to? Yeah.
I'm not sure I understand the term fixation. As of clips, I get it, but the first term, I'm not sure I completely grasp.
So this, I should have mentioned, these pluses, we just, between clips, we have a screen with a plus where we just tell people to fixate, to look at that and do nothing. And it's just for technical reasons, usually because we're actually measuring blood flow and not electric activity, so we need to separate things in time. It's very slow, so if we want to see a response, we need to separate it from the next response. But then this technical issue, we suddenly find it has real cognitive meaning. We need these fixations. And that's actually something interesting that we find here. Because having 2 clips one after the other, it reduces the response in the hippocampus to the first clip. If you're doing something else, anything else after the clip, then you have less of a response to the first clip than you would have otherwise, and your memory goes down. And if you think about it, in real life, if, let's say, someone is giving a talk and talking too fast and not pausing, you can't really grasp what's going on. And that could be because we need these pauses in order to get the best activity that we can.
If we measure the time, the length of the clip, the maximum length of clip that is available for memory, because you mentioned a clip, but I was thinking of a movie. If you watch a movie of 1 hour and a half, it's actually many clips. So what is the maximum time for maximum peak?
So first of all, perfect. That's just going to be what happens in a whole movie is going to be the next slide. But in terms of the maximum time, we looked at that. And it seems to be that around maybe 16 seconds, 20 seconds, you'll already start to see something going on. There's a limit to how long you can keep something before you save it to memory. And there's probably a hierarchy where you save small chunks, and then larger chunks, you kind of bind them together in larger chunks. So, but you do have some small unit that you must encode. Yeah.
The length of the clip and the length of the activity after the clip are somewhat similar in the 5 to 10 second range. Is that coherent with what you see for longer things as well?
So we looked at that, whether we'd see longer activity for longer clips, or maybe stronger activity because there's more information that we're encoding. We don't really see that. The length of this response is determined by what the blood flow looks like. So this response starting at the end of this clip, that is what you'll see in terms of the pattern of blood flow, even in response to just a flash of light. Something like this, it takes several seconds. And anything, just because we have very fast electric activity in the brain, but it takes time for the blood to flow. So no matter how fast, how salient the activity, it takes some time to send blood to that area in response. Yes. So it does seem, the pattern is the same, but we do see more that people remember these emotional clips more, and then we see stronger activity. But let's say you have a completely non-emotional clip that everyone remembers. For some reason, there are some clips that everyone seems to remember, then we'll also see the strong activity. Even a super boring clip of someone laying on the grass. I don't know why. Yeah. Originally, yes. Usually in fMRI, you separate, depending on what you do exactly, you separate every event from one another with some fixation like this. So what does this reflect? We think that perhaps we experience something, we keep it in some temporary storage, and then when it has meaning, when we can wrap it up as a unit, we wrap it up and save it to memory. And again, Inside Out very nicely demonstrates what we think is happening. And I think it's nice that the science kind of led us back to something that's actually very intuitive, and it's how people think about memory, but it's completely not the way scientists thought about memory for a long time. So you see here, they're having experience quite like my own in daily life. And then after each such experience, we create these orbs, these balls of memory that represent our experience. This is what we think could be happening. But what's the end in real life or in a whole movie, right? We don't have ends. I'm not pausing and showing you a gray screen every few seconds in my talk. And so for this, I'd like your help with a little experiment. I'm going to use Birdman, which is a movie that's one shot. There are no cuts in this movie, so it's very nice for demonstrating this. It's quite impressive. And what I'd like you to do is to clap your hands when you feel that one part has ended, one part of experience, one unit that has meaning, and something new has started, even though there are no cuts. Let's see if it works. It's harder with films like this, but we'll try. Ready? Okay.
You don't try to kill people.
Yeah, good. I don't know. What do you think, boss? Okay. You want to do this with me?
What do you think?
Okay, not that many claps as usual, but often people feel something new has started when a new person enters the conversation, right? It's no longer a conversation between these two people. Now someone has entered, something new is happening, and probably they're going to change what they're talking about, things are going to change. And so these moments in time where people experience an end, even though we go about life, it's continuous. Often people experience these ends, these boundaries between events at very similar moments in time. And so these event boundaries cut up our experience, and they could potentially be the moments in time in real life where our hippocampus says, okay, let's wrap everything up and save it to memory. And this is what we tested. So we wanted to see whether the hippocampus really responds at boundaries when we show people films. So we took people watching a Hitchcock film and people watching Forrest Gump. The black line here are moments in time where other people, it's not even the same people who we measured their brain activity, completely other people said, oh, this is a boundary, here there's a boundary just like you did now. We took those moments in time and we looked at the brains of other people being scanned while they were watching the films. And we see this beautiful response in both films, and it's even stronger for stronger boundaries. So when there's a significant cut, your hippocampus says, okay, probably something new is happening now. Let's wrap up everything that just happened and start a new event. Start encoding something else. So we see that it really responds with boundaries. So here, this study had a lot of different age groups, and we focused on young. And if you ask my definition of young, it's everyone my age and younger, so I couldn't put myself in the old group. But another group, after we did this, looked at the older group as well, and they see something similar, but a bit of a reduced response. But there's a similar pattern, and very recently, amazingly, a student of mine showed me scanning of, I think, 2 to 9 month old babies, and they have responses at boundaries. So they're kind of different boundaries. They can't be boundaries due to meaning, change in topic of conversation, but if there's a significant shift, they already see a response in their hippocampus. So it's something very, very basic about how we interpret the world. So, yeah. So that's something we want to test. We haven't tested yet. Not for a whole lecture, perhaps, but even in these clips, you can sometimes have an event that has closure, right, that kind of wraps up, or like PowerPoint plays some tricks, which was, of course, fine this morning, and now cut the movie before the end. Will that create a lower response in the hippocampus because the clip kind of got cut in the middle? Or will you actually have a larger response? It's a good question we still need to answer. So you and, yeah. Very
While the hippocampus is wrapping up the memory and trying to form it, did you test if the thing that happens after is kind of like not remembered well?
So it's the opposite. If we have two clips, one after the other, the second clip is remembered perfectly well, even a bit more than a single clip, maybe because you're already engaged. But the first clip, because the activity is at the end, it's not the first clip that's disrupting the second, it's the opposite. The second clip is there while you're trying to wrap up your previous experience. So it's kind of reducing that response. I think it's more, yes, when you have experience, then you're engaged in that experience. It doesn't even have to be another clip. I can tell you to count backwards, and that will even reduce your memory and your response. Anything you do, immediately after. Yes?
I think the sociologists say that the maximum attention span without interruption is 20 minutes. Have you tried to make these experiments at the beginning of a 20-minute speech or at the end of a 20-minute speech? And is there a difference in the memorizing depending when your attention span was at minimum or maximum?
So we do see some patterns over time, but they're not consistent across all people. So some people will be especially attentive at the beginning, some will be especially attentive at the middle or the end. We don't necessarily see a certain pattern of 20 minutes and then they zone out. And some people have incredible memory. I have no idea how, they just remember everything. Need to study their brains especially well. So since then, there really have been a lot of supporting findings in rodents and in electrodes implanted in humans and a modeling study. So it really is becoming more and more convincing that this is how the hippocampus works. And this brings me to the main question that is now leading us, that we now want to answer. And that is, what is this unit of memory? So if we're encoding at boundaries, what are we encoding? And we can think of two general options. One, if I have this experience from Cambridge, let's say, every moment I experience, I'm saving it to memory as we go along, and maybe we have some additional activity at the end that does something, but we actually encode every moment in time, and only afterwards our experiences become, let's say, you bind together one slide of my talk with the next slide because they were in the same room or something like that. But what we're trying to suggest is that as we go through life, we're kind of aggregating information, keeping it for a moment. When it has a meaning, when it's an event, we save it to memory. When something new starts, now something new started, we're keeping that until something new starts, and then we're saving that, and so on. So we're saving entire chunks, entire events. This is what we want to try and understand. And so more broadly, we want to understand how these dynamic events, these experiences, how they're encoded. So are they encoded, tac, tac, tac? Are you encoding every word? I'm saying it doesn't make sense, right? You're not saving now to memory word by word. Or are we wrapping things up? And it's not enough to ask this. We also ask what the boundaries are. How do we know that something new has started? How do we know we need to now encode? And then how do we represent these events? How do we retrieve them from memory? And I'll talk a little bit about the directions for each of these, starting with encoding. So in this study, Kevin Campion, a student of mine from Cambridge, created a 40-minute film using the computer game The Sims. Okay, this is a completely artificial film where we had different levels of experience. We had the characters performing different actions. Then they could jump to a new scene that was in a very different location, and a completely different character. So this gives us different level of boundaries, and I'll show you an example to demonstrate. So these, for example, are the actions. This is a single scene. So she does push-ups, and then she goes to this weight machine. And now we have these transitions, let's say from being inside the gym to being outside in the dressing room. Then a larger shift from the dressing room to being in Starbucks. And finally a shift between Brandy, that was Brandy, and this is Greg Greybeard. They all had names so people would be able to remember them. And now what we want to do is see, first of all, whether in this continuous film we can identify a response to actions and the different levels of boundaries. So when we look, we see here that there's no response to camera changes. So it's not that the hippocampus is just responding to a shift in the camera. People are oblivious to that. We see a small response at the ends of actions, which we weren't able to see before. By using these very carefully crafted stimuli, these carefully crafted movies, we can separate each action from the other. And now we suddenly see that the hippocampus is responding even to these very small units. But there is a much larger response to boundaries, these transitions between scenes with a stronger response to stronger boundaries. But is this linked to memory, right? That's our goal. That's what we want to understand. So what we're asking here, if we have this film, and we know we have this response at the end of the action, and we have these increasing in strength responses to boundaries, what is going to predict whether you remember that she did push-ups? Which activity? Is it going to be this activity at the end of the action? So each action is encoded in itself, and we see stronger response at the end of actions that you're going to remember, or will we see at the end of a scene a stronger response if you remember more of the actions in the scene? And when we look at the results, we actually see a response to each action. Because we separated these actions so much in time, we now see that they can't be a single event if they're spread out in time. Because you saw she did push-ups, and then she walked over to a different, in time and space, she walked over to a different location, and there she did something else. So for now, we actually see the unit of encoding is smaller than we thought. It's already at the end of a single action. And when we look at the end of a scene, these responses are quite messy. We see some gradient with a stronger response if you remember more actions, but it's much less convincing. I'll be able to tell you, I think, in about a month or two, whether that is convincing or not. We're still looking at this part. So overall, we see quite a small unit of activity. But can we have, now we want to ask if we have the actions one after the other, which is more similar to real life, can we see encoding of a whole sequence of actions as one unit, which matches what we feel or think may be happening. And for this, we started a collaboration with the Hadassah Medical Center. There are people with intractable epilepsy that can't get better just with drug treatment, and so they have surgery. And during the surgery, they have electrodes implanted in their brains, and they spend around a week at the hospital in order to monitor their brain activity. And often they have electrodes in their hippocampus, because that is an area that tends to have a lot of epileptic activity. So we're collaborating with them, and now we have direct recordings from the brains of kind of healthy humans. They have epilepsy, but we can see direct recording from the hippocampus as people are learning things. For this study, I don't have the results yet, but I do want to show you the concept of what we're doing. So we have these Sims clips, like the one I'll show you, and you'll see that now the flow is a bit faster. So we have this action, he's lighting the fire, and then he's going to sit, let's say. And what we think will happen here is that the hippocampus will wait until the end, and only this activity in the end will tell us whether they remember that he lit the fire, even though he went to sit afterwards and more things happen. So we want to see if this time the hippocampus will really wait until the end. And we have other clips, and this is what's great about the Sims, where you can see, look at the bottom clip and see how it differs from the top one. You can see that they're identical, right? The Sims gives us the ability to switch a person and switch a location, but keep the clip identical. And now we predict that we'll see this brain activity that predicts memory for the fire much, much earlier at the boundary. And by directly testing from people's hippocampus, we can test this and see whether indeed the brain activity really is at the boundary. So this, again, I'll be able to tell you about maybe at the next brain circle.
The advantage now is that because you have an electrode in the brain, the time resolution is much better than before. With almost blood,
Yes.
it takes a second to get to the region.
Mm-hmm.
The capability to look at much shorter.
Exactly.
Because you record electricity rather than blood.
Exactly. We have the ability to distinguish between brain activity that happens at a certain moment in time versus 1 or 2 seconds later. And that's key here. Okay, so that was a little bit about encoding. And now we want to ask whether when we remember things, we also remember them as a whole unit, right? If we encode things as a unit, encode an entire event, then we expect that if you're retrieving something, you're also going to retrieve it as a whole event. And so another study by Kevin Campion, he presented people with clips like these.
Quick scrub. Connoisseur. Takes effort. Truly adorable.
Okay, so he has these clips. Each one has 2 actions, and each action is associated with a phrase. And the participants are told that these are the things the actor performing the actions is thinking to themselves in their mind, and they should remember them. So we have these different action phrase pairs. And now, after we have several videos like this with scenes, and after every one, we ask people to reactivate memories. What do I mean by reactivate? We ask them, what was the action associated with the phrase connoisseur? We just ask them to try and retrieve. We don't tell them if they're correct or not. We don't tell them the right answer. They just try to retrieve it. And they have this 6 times, videos and retrieving, reactivating. And it's known that when you do this, you really improve memory for what you tested yourself on. So this is general good tip. If you want to improve memory for something, test yourself. Rather than repeating it, just test yourself and it improves memory. And so afterwards, we have a test and we ask, which action was associated with different phrases. So we have what we call reactivated phrases, like connoisseur. They're reactivated because we ask them to retrieve them. So we know that these are going to have a big improvement in memory later on. We have the non-reactivated. This was reactivated, connoisseur, takes effort. It's 7 seconds from it, but this was not reactivated. Okay, so we expect to see, and we do see, a much larger, much better memory for connoisseur compared to our baseline here is the not reactivated. So we have much better memory for the actions associated with connoisseur compared to the action associated with takes effort. That make sense so far? And now what we want to ask is whether when they reactivated connoisseur here, did they automatically reactivate the entire event, even though we didn't ask them? So are we also going to see better memory for quick scrub, even though it's also 7 seconds from the action we reactivated? The direction could be either before or after, okay? And so we looked at the reactivated event, something where it wasn't reactivated by the event was, we see a smaller increase, but we still see a significant increase in memory. So that means that when they were thinking of connoisseur and we didn't ask them anything about quick scrub, the whole event came to mind. So it means our memory is organized in this way as events, and if we bring back to mind one part, we're going to bring back to mind everything that's stored. And it goes even further. We ask, what is the event being reactivated? And because we have this beautiful control with the Sims, if this was the original film that I showed you, and let's say this was a reactivated element and this was, we can now take the scene and and this was a reactivated event, and this was not reactivated. We can replace this scene with a non-reactivated action with an identical scene, except now it's the same character and the same location as something that was way earlier or way later that was reactivated. And now the non-reactivated one also gets a boost in memory. So it's not just the event itself. When I asked you to think about breakfast this morning, you probably also brought to mind, even if you weren't aware of it, yesterday's breakfast. So you improved memory also for breakfast yesterday, and not just for breakfast today. Now everyone is going to remember breakfast here very, very well. So it seems that the event can be in memory, doesn't have to be continuous. And maybe you can even have it over a long time. Maybe you're binding together one talk with another talk of the same person that you hear a month or a while later. And I do want to say just one word about brain activity. I'll give you a small example of the directions where we're looking at what the brain activity looks like when we're retrieving these events. This is a whole line of work, but I'm just going to give you one small taste of this. We had the same clips presented over and over again. So until now, I showed you only new clips where you didn't recognize anything. But here we have clips that repeat, and we have clips that were presented only once. These are single presentation clips. And now we look at the brain activity in the hippocampus. First of all, only for the single presentation clips, and this is the first presentation of the repeated ones and the single presentation clips. And we see what we saw before. And now if we add repeated clips, I'm adding here presentations 4 and 6, we get a very different pattern. Focusing on the end, we see that once it's familiar, you don't bother encoding that much. The response goes down. I know that. I don't need to work that hard to encode it. I already have some representation. But what we also see, at the beginning of the clip, you see this small jump? We suddenly see something that looks like, I remember that. I saw that. So the hippocampus kind of retrieves the event at the beginning, and this aha is what we're focusing on now, trying to understand what that aha includes and whether it includes the entire event. So again, stay tuned. Yeah. So deja vu is a bit different because this is something you actually saw. I don't know if we had deja vu without actually, where in deja vu we never actually have the experience. I actually don't think we'd see this response. There are different regions that are more involved in a sense of just something is familiar. Usually the hippocampus is involved more in bringing everything back to mind. So here, you're not just seeing that the clip is familiar, but you're lightly bringing it back to mind. You know what's going to happen, right? You kind of play it very fast in your mind. And if we don't have something like that, I don't think we'll have this response. Although sometimes in deja vu, we feel like we're going to predict the future, right? We know what's going to happen. Interesting. So whether, because we know that familiar environments and familiar places are calming, could this be mediated by this response? So there's a big debate if the hippocampus really is involved in just the familiarity, or it's reflecting the fact that you're bringing everything back to mind and a different area is involved in familiarity. So here too, I would think this is probably not directly involved with the calming effects of a familiar environment, but rather this is a specific aspect of the familiarity of bringing everything back to mind. The episodic memories, retrieving them, is thought of as mental time travel. You project yourself back to when you first experienced it. And you also. So my guess is that a multimodal, so whether we'd see also, whether this is multimodal and how that would affect the response. So I'm guessing whatever the trigger is, if it elicits a whole memory, if you really, the smell brings you back, like in Ratatouille, did you see Ratatouille? So if it brings you back to your childhood, then you're going to see this. If it's just, that scent is so familiar, but I have no idea where it's from, then I don't think it's going to elicit this. And if the original memory is richer and more full of details, that's probably going to give a strong response. So if you also bring back the smell and the sound, then I imagine, but again, this is all guessing, okay, based on our previous work. We haven't shown it yet.
Manipulate performance by giving instruction to people when they watch the clip.
Oh.
If I say, just watch the clip and enjoy, or if I say, please pay very close attention, it's going to be a test. That's a huge difference.
Okay, so I should have added this study. There are two things relating to that. One is we ran pretty much that study, actually in two different ways, and we told people, just watch these clips, but be attentive, or you're going to have to remember this. Any guesses what the hippocampus does when you tell people you have to remember this? What? Oh, forget something else. Interesting approach. It's kind of what happens. We suddenly see that is not the role of the hippocampus at all. There is actually no difference anymore between what you remember and what you forget in the hippocampus, because what determines that are other regions in the brain. So this is the natural processing. What we're really trying to do, and we haven't gotten there yet, is to move the boundaries around with instructions, to tell people, now 6 pictures are a unit, or now only 2 pictures are a unit. And we're still working on that. Yes?
I'm assuming, just let me know, right? When you dream, you don't see that signal.
I don't know. Yeah.
Why I'm waking up, I remember my dream, then I'm not remembering.
I'm afraid I do not know the answer to that. One of the issues is that we don't know when this is happening. So here we know when you're presenting it, and then we can look at the signal that's related to later memory. But in the dream, we don't actually know when it's happening, and it's so condensed in time. There are studies starting to look at lucid dreaming that Hagai mentioned, telling people, teaching people how to do lucid dreaming, and then talking with them during sleep, which is incredible. They actually tells them, move your eyes to the right or to the left to answer yes or no. And then we can start to look at that, but it's still a bit of a long way before we can actually look at these things. Oh, yeah, I'm kind of running. But we started late. Yeah. Okay, because I was getting the enough. Okay. Okay, so a short word about how we segment these events, okay? And if we look at these boundaries, and we want to understand what they are and what causes them, then let's look at this. We have, they're going to continue talking, and then, someone entered. And a lot of things are happening at this moment in time, right? We have surprise. We weren't expecting someone to enter. We no longer really know what's going to happen, because before we were just assuming they'd keep talking, but now maybe they'll get up, right? Maybe it's a wave to it. We don't really know. There's a change. A new character came. And there's a new story. And we want to understand what exactly makes a boundary. Which of these is triggering the response in the hippocampus? And again, we're starting to look at this. And specifically now I'll talk about trying to understand what are the effects of change as opposed to a new story. Currently not yet on the hippocampal activity. And just like there are a lot of characteristics of boundaries, there are also a lot of effects of boundaries. We see changes in brain activity all over the brain. We see that your eye movements change, your pupils change in dilation, memory changes, your reaction time changes. These boundaries are a very important organizing principle of all of our experience. And we want to try and understand which of these effects is really related to what we define as a boundary, which is a new event, a new conceptual event. And so we're going to here focus on two effects on memory that are taken as standard effects that characterize boundaries, that people, when they see them, they say, this is a boundary. So in many studies, I'll give an example, you can also have boundaries, not in films, but just with these very simple objects. But let's say you suddenly change the color of the frame. This is also a boundary. And people see, for example, that if you have what's called an associative memory test, if I show you the shovel and I ask you, was it surrounded by a purple frame or an orange frame? And I compare a boundary item to a non-boundary item, what do you guess? Where would memory be better? For the boundary, right? Because we're more focused, we're kind of oriented, something new, let's pay attention. And if we ask which came first, we can look at two items within an event or two items across an event, the same distance, and we ask which came first. Any thoughts? What would be better within an event or across event? Within, across. I thought it would be across. It's within. We have much better memory within event. And these are taken as standard indicators of boundaries. But like we said, boundaries have change, and they have surprise, and they have so many things. And we want to understand what is linked to the new story, the new event. And this is part of a much bigger study, but I'll just mention the part relevant here. We had, for different reasons, these were in circles instead of squares, but we had these events very similar to the study I showed you. But we trained people. We taught them in advance that there were different types of events. Some events were like the ones before. There was just a blue event called the butterfly event, but we also had the rose event, which was alternating between pink and green. So now here we have the parasol event, and here we have, for example, the watermelon event. And now we have the regular boundaries as before that include also a change in color, but we have boundaries that you know something changed because nothing changed. So because you have the same color, you now know You're in a new story. You're in a new event. And the same with an event. Some have alternating colors and some have the same color. And now we can see if these measures are actually really due to a change in story. So when we look at dissociative memory, we see that it's actually not sensitive at all to boundaries. We have a stronger response. These bars are higher when there is change, and a lower response, these light bars, when there's no change. It doesn't care about boundaries. So it was actually never a marker of boundaries. It was also always about the color change. But when we look at temporal order memory, now we see overall a stronger response within and a weaker response across, and we don't see a significant effect of change. So this is really due to the fact that the event changed. Okay, now we can start to dissociate all these different effects and take this to the scanner and look at the hippocampal activity. So what would I like you to take home? There were a lot of different things, but first of all, I'm hoping that if someone now asks you what your favorite region of the brain is, of course it's the hippocampus. I mean, it's a seahorse. I pity the people who study the amygdala. It's an almond. What can you do with an almond? And the main thing is that the hippocampus really responds at these boundaries, at these moments in time where something new is starting, and that's what predicts what you're going to remember. And that may reflect the forming of the stories of our lives. Perhaps this is when these stories that we get to keep about our lives are formed. And the experience is that ongoing experience that we keep losing because it's just these moments that fade away over time. So maybe this is where memory is being saved. And mainly I'd like to leave you with a question more than an answer of what are these molecules of memory. And I'd be very happy to hear your thoughts, because as you saw, one of our biggest discoveries came from just looking at our experience and understanding that, and then finding a way to look at that scientifically. So I'd be very happy afterwards to hear your thoughts. And I've shown you how we're starting to reveal the answers to this, and mainly the approach and the question. So why does this matter? First of all, it all starts to fit in, right? If encoding takes place at boundaries, then we suddenly understand why we need these breaks. And we can start to intentionally insert breaks in the right places. If you're in a meeting with someone and you want them to keep important parts, and you have to pause after those moments. And it's been shown that if you put commercial breaks at event boundaries in a film, you're going to improve memory for that film, despite the fact that you're adding these commercials. So we want these breaks, we want these shifts. And we can also start to use this for education to understand what the best units are. And are these units different for people of different ages? I'm guessing there are generational differences. And I'm not talking about older adults, younger adults. I'm talking about TikTok generation. Because they're used to getting information that's in tiny, tiny units and comes very, very quickly. So maybe their basic units, some people here probably have smaller units than I do. Maybe the units change. And even though this isn't directly related to memory disorders, if we want to start to understand memory disorders that affect people in real life, we can't just be focusing on understanding how we form a memory for a picture or memory for a word, right? We want to understand what memory is built of. It's a basic theme in science that if we want to understand something, we first break it down to its parts. If we think about material, we want to understand the molecules. If we think about the genetic code, the language, I know it sounds ambitious, but we want to do the same for memory, to understand what particles, what units it's built of, and then we can start to understand and test, let's say, memory problems in a different way. And before we end, I've shown you kind of what we do with The Sims, and it's very nice, but we're starting to play with AI. So just to show you what we're doing with this, it's still at early stages, but if you look at this clip in the middle, you can see how we can change the person or change the location using AI but still have them do the same thing. So this gives us very nice control, similar to what we could do with the Sims. It's not without its issues. We have our greatest misses directory on the computer. So for example, this perhaps is not a very natural, I mean, we'd all like to have a third arm and levitating balls. But if you notice these two films, if you didn't notice anything odd about them, we generated these with AI. There weren't actual actors, these films that I showed you before. The capable. Yeah, so they're bad actors, I agree. But if I showed you some of the original films I used, which were student films, they're kind of also similar in how they're acting. It's not completely natural, but it's quite amazing what we can do with this. So this is where we're going. And with that, thank you. And I'd really like to have conversations and hear your thoughts afterwards. Thank you.
Question?
Yeah.
Where is the other one? There was another one.
Sure.
I was just going to ask, do the breaks in between the segments have to be a stimulus, or if you just showed like a black screen, would that act as the same?
So I think the fixation cross is really very similar to a black screen. The reason we use that is it's actually less salient than having a black screen. When you're watching a movie, if you put a gray screen, it feels more neutral than suddenly switching to black, especially if the movie is bright. But I think the less stimulus there is, the more you're going to see. I actually even saw, I analyzed some data set where there were black screens, and it was the same.
Have you tried across different cultural people? Do they react in the same way to the stimulus?
So the hippocampal response seems similar, but on a very small sample, 8 Germans and 8 non-Germans, we had, I'm trying to remember now if it was much more boundaries or fewer boundaries for the Germans, but there was a difference in the number of boundaries they put. That's anecdotal though. The Forrest Gump study was actually, the film was in German, so we brought German speakers.
You're referring to activity in the hippocampus, but do you know what's happening there inside? I mean, how does it fix?
So that's one limitation that we have when we study these things in humans. We can't look at the changes in the synapses. We can't look at the structural changes, the implementation. And that's why I think there's something really beautiful about LSEC, because it's not enough. I love studying humans. I'm interested in episodic memory, and I think you can't really study this in animals. But it's not enough. We need to have all the levels and then see how they connect together. So to understand how we go from this to understanding how exactly the wiring changes, we need to go to studying something complementary in animals. We can go into a higher resolution and look not just at the activity, but look at the precise patterns. I don't want to get into, I'd be happy later to get into the exact explanation, but one thing we're looking at is to see, let's say, whether a very specific pattern represents this clip and a slightly different pattern. So one point in the brain is slightly higher, one is slightly lower, that represents another clip, whether we can really see activity that represents each and every clip. But I'd be happy to elaborate later.
Maybe I didn't understand everything, but I want to ask you, have you studied different emotions
So
when somebody answers?
Oh, and the answers. No, we haven't looked at the emotionality of the answers. We did in the first study look at emotionality of the clips, and we did see effects, but those effects were due to the fact that the emotional clips were more memorable. So if we looked at very memorable clips that were not emotional, they were the same as the emotional memorable ones.
You don't analyze, for instance, if somebody is sad when he's answering?
No, we don't look at that. But that would be an interesting direction to see how general the general state that we're in affects what we're doing.
I think we should take a little break. There is a coffee there for 10 minutes and then come back here to listen to Avitan.